Formation of the neural plate. By the 18th day of embryological development, the notochord, situated beneath the ectoderm layer, sends inductive signals that stimulate the ectoderm to thicken along the midline. This process forms the neural plate, a flat, elongated structure resembling an inverted pear. As development progresses to the 19th and 20th days, the neural plate begins to undergo significant changes. It elongates, becoming narrower, and its edges thicken to form the neural folds. These neural folds rise gradually and move towards the midline, initiating the process of neural tube formation, which will eventually give rise to the central nervous system. Closure of the neural tube. The neural tube closes in a cranial to caudal direction, completing the formation of the early central nervous system. As this occurs, three primary dilations, or vesicles, develop at the cranial end of the tube. Prosencephalon. This region will differentiate into the cerebrum, thalamus, hypothalamus, and other associated structures. Mesencephalon. This structure remains as the midbrain in the mature brain, responsible for processes like vision, hearing, and motor control. Rhombencephalon. This area divides further into the metencephalon and the myelencephalon. Meanwhile, the caudal portion of the neural tube elongates to form the spinal cord, which will facilitate neural communication between the brain and the rest of the body. Neural crest cell formation and migration. Once the neural tube is formed, neural crest cells separate from the neural folds. These cells undergo an epithelial to mesenchymal transition, where they change from tightly bound epithelial cells to more migratory mesenchymal cells. This transition enables the neural crest cells to become highly mobile and versatile. The neural crest cells migrate to various regions of the body, including the pharyngeal arches, where they contribute to the formation of diverse structures such as bone, elements of the facial skeleton and cranial structures; cartilage, supportive frameworks within the pharyngeal arches; nerves, peripheral nerves, and ganglia. This migration is a pivotal step in the development of numerous tissues and organs,